Associate Research Scientist at Yale University, working with Dr. Sepulcher in the Radiology and Biomedical Imaging Center. She completed her bachelor's, master's, and PhD in Spain, and her research is pretty broad. She's published on a lot of different things, but a lot of her work focuses on AD pathology and using neuroimaging. Today, she's going to talk to us about integrating spatiotemporal multimodal neuroimaging using graph theory. And I know a few people here are starting to use graph theory, interested in it, so I think it'll be a really cool talk. Okay, go ahead and take it away. Actually, just one question. Do you want questions at the end or during the talk? Do you have a preference?
I'm fine with whatever you prefer. Yeah.
Okay, so we'll plan to save things for the end, but if you guys have a clarification question or anything like that, feel free to come up and pause and we can ask that to make sure we all know what's going on. Okay, thanks. I'll go ahead and mute on our end and you can go ahead and get started.
All right. Thank you for the introduction and the kind words. And hello, everyone. And also, I would like to thank Ohio State University for inviting me today and for the organizers of this series as well. I am going to talk about graph theory, and I'm not very sure about your background in using graph theory. So, okay, yeah. So I will start talking about the basics of graph theory, and then some of the measures that we use are very focused in centrality and segregation and integration. So I will be mostly focusing on that, but graph theory is very broad, and you can, of course, use many measures to apply to your neuroimaging studies. Then I will talk about multimodal integration. And finally, I will present a couple of projects that we are doing in aging and Alzheimer's disease. Okay, so what is graph theory? In neuroimaging, graph theory provides a framework of knowledge for the study of the topology of the brain networks and their temporal and spatial organization. When speaking about graphs, when speaking about graphs, we usually represent the brain regions as different nodes, like you can see here, and the relationships that we are measuring between these brain regions are expressed using links or edges. As you can see in this figure, nodes and links can have the same size or different size. And this is more for visualization purposes, but the size that we give to the nodes and the links usually represents the relationship or the importance that the nodes and the links have in our network. And everything about this is recapitulated in a matrix. We call this matrix the association matrix, and normally we arrive to this matrix by using correlation analysis, although other kind of statistical measures could be used. If we, for instance, think about the brain is parcellated into 6 big regions from A to F, we will represent these regions at the edges of the matrix, and then we will represent the relationship between the regions inside the matrix. So in here, for instance, if we have node A, and we want to know how it's related to the rest of the network, we will see, for instance, that it is related to C, and the relationship between A and C is denoted by a 1. Sorry, can you see my mouse?
Yeah, we can see the mouse.
Okay. And then whenever there is no relationship between two regions of the brain, then we use 0. All in all, we can take back this association matrix into a network representation, and then we will draw our network with our 6 regions of the brain, and then the relationships will be corresponding to the links. So we say that between A and C, there is some kind of relationship. So we see a link in between them. And between A and F, because there is no relationship and we put 0, then there is no link. And what is connectomics? I think about connectomics as a group of computational solutions based in graph theory to resolve specific questions. One common particularity of the brain is that regions that are anatomically separated show similar structural, functional, or molecular properties. And connectomics help us understand these properties by investigating the relationships. For instance, imagine that you are doing an structural research project and you have the images of the brain of several individuals, and you are measuring, as I said, anatomical properties, and each of the regions of the brain will be corresponding to one voxel. So we painted all our brain in blue and imagine that we have several dots or several nodes representing the voxels of the brain. You can take the volume, the anatomical volume, and represent it in a matrix form in which each of the voxels will be located at the edge. And then because you only have one image per participant, each of the participants will be located at the longitudinal edge. And then if you want to build your association matrix, you can perform a correlation analysis between the voxels of the brain across the participants. In this way, you will obtain a brain matrix. And you could, from here, do group level connectomics. If we go back to our network representation from the association matrix, you will have a huge network because you have many voxels in the brain, and each of the voxels is related to the rest of the voxels. And because there are a lot of them, we cannot see the links in between them. So how will this look in real? So in here, I have one of Jorge, Dr. Sepulcre, investigations in which he was actually researching how the human brain integrates the information from external sources and internal cognition to produce a coherent experience. He was using resting state fMRI, and he had several images from different individuals, and he wanted to know how the information was traveling from one specific region of the brain to the rest of the brain until it was converged into something that we usually call higher functions, higher cognitive functions. So again, we have the voxels of the brain. And then in this case, because it's a time series, because it's functional connectivity, we have the time. And if we compute a correlation between the voxels of the brain across the time points, we will obtain an association matrix in which we know that each of the voxels is more or less related to the rest of the voxels of the brain. If we take the association matrix to the network visualization, what we will see is a bunch of nodes that are represented by the red dots, and the links between the nodes are represented by the edges or links between these regions and are in black. So let's go now to centrality and how we can use the association matrix to learn more about the relationships between the nodes. So we have several centrality measures, but I'm only going to talk about centrality as degree centrality. So what is degree centrality? Degree centrality is the sum of the correlations of the connections of a node across the entire brain. So again, imagine we have a more simple network and we are investigating this node that we are going to call I. We are going to call it I, and we want to know how important is this node in the rest of the brain. We will use degree to do a sum of the connections of I with the rest of the brain. And then if we actually do the computation, we will say that the degree of I is 5 because I is connected to 5 different nodes. On the other hand, if we want to know how important are the relations between I and the rest of the different nodes, instead of doing a binary computation, we can actually look into the correlation values. And then we will talk about weighted degree. So weighted degree is the sum of the weights or the sum of the correlations of the node. In this case, it will be 1.6 because what we do is just sum in 0.3, 0.3, 0.4, 0.3, and 0.5. Only the direct relationship between I and the rest of the nodes. It is also important to know that whenever we use a matrix that is not symmetrical, and it contains information about, for instance, temporal dynamics, we can also know how the information travels from one node to the other because we know how the relationship change in time. Whenever we are speaking about information that is traveling from our node I to another node, we talk about out degree. And whenever we are looking into the information that is coming into our node, then we are talking about in degree. So out degree will be information that is going outside of our node, and in degree is information that is coming inside our node. And these relationships normally are not static. Normally they are completely dynamic. And the interesting thing is that if we use graph theory and we actually use several imaging modalities, we can integrate all this information and have a more comprehensive understanding of how our network behaves in the space and in time. Finally, of course, networks are very complex in the brain, and we use other metrics to integrate all the information into something that can explain what we are observing. Imagine that our network is much larger, we have more nodes, and whenever we see that there are different modules inside the network, we are speaking about segregation. As you can see here, there will be 3 different modules: the green module, the blue module, and then the orange module. But we also know that the brain doesn't work in compartments. It actually integrates all the information, and graph theory is able to actually locate which are the integrators or the regions of the brain that allow multiple systems to communicate between them. So segregation usually denotes systems functional specialization, and we observe segregation in discrete systems within a network. To measure segregation, there are multiple modularity metrics that I'm not going to discuss here, but if you want to look into more, if you search for graph theory modularity, it will be easier. So we observe segregation when we are looking into primary systems. For instance, we have the visual system, we can have the motor system and the sensory system, and we have the auditory system. Integration, on the other hand, are the systems that allow us to actually converge the information and actually use it as something meaningful. We have the multimodal areas that usually are close, spatially located to the primary or sensory areas. And then we have also the higher or more higher cognitive areas located in the cortical hubs. And at the end, what we are using is graph theory to locate which are the modules of a network and which are the integrators of these different modules. Let's go to something more specific to look at how graph theory allows us to use and to understand how the brain works. So in one research that we were doing several years ago, we were trying to find out the visual motor integration system and how this system is actually related to different disorders, developmental disorders that present problems related to visual motor integration. So in the first part of this research, we use graph theory to actually locate where is the visual motor integrator area. Imagine again that we have our brain and we have our network in here, represented by all the different nodes. And we know that some of the areas of the brain are specialized in processing visual information. And here we are representing these areas in yellow. And other areas of the brain will be specialized in motor processing, which are represented in green. And what we wanted to find is where are the integrators of the visual and the motor cortex or motor integration information. So in this little toy model, we have in A, the visual areas, and in B, the motor areas, and C is what we wanted to discover. So we use neuroimaging, we use task fMRI, and then we use task connectivity and resting state connectivity to define where is the visual motor integration system. With task activation, what we wanted to do is actually to really make sure that we were measuring where the different primary visual areas implicated in a specific visual motor task were located and where were located the motor areas. However, task activation doesn't really tell us anything about how the areas are related in the brain. So we couldn't find out the red nodes. So we use task connectivity and resting state connectivity to research the relations that were behind the activation of the different nodes in the primary visual cortex, the primary motor cortex, and how these two different modules of the brain were integrated. And finally, what we obtain is a map of the visual areas, the motor areas, and we discover where the integrators were located. So as I said, in the task that we use in here was a finger tapping task. And with weighted degree, we were able to locate the areas that were more activated during the task. So we just did comparison of the activation blocks against the control blocks or the fixation blocks to locate motor activation and any kind of visual activation. And then we use task connectivity and resting state connectivity to locate first with weighted degree the activation of or the functional response of these areas. And then we use a stepwise connectivity to actually locate the relationship between the visual areas and the motor areas. So stepwise is a metric that allows us to investigate direct and indirect connectivity. And I'm going to go back one slide to explain this more. So imagine that we are in our seat region located in the visual cortex. And for instance, we choose to start our metric from the cortex, from the region that has more activation. So what we do with a stepwise connectivity is to relate first the regions that are directly connected to our seat region. So we will locate this region, and the direct connection between this region and the rest of the brain will be other two regions that are located in the visual cortex. And then with two-step links or two link steps, what we do is to actually locate the regions that are connected to our seat region, but are not directly connected. They are indirectly connected. So we will go one step and then the second step. And we do this in a way that we introduce in our code, okay, give me the connection first of the region of the yellow region that is direct, and it will only locate regions that are at one step. And then we introduce our yellow region, that is the visual, and our green region, that is the motor cortex. And we will ask the code, now give me the location or the relation between the yellow visual and the green motor that is 2 steps away. And it will give me the regions of the brain that are functionally connected or functionally related. They show similar activation. And in that way, we will be able to locate the regions that are actually acting as integrators of the visual and the motor. So we did that at the voxel level, and we found different regions in the brain. And of course, we found regions located close to the visual area, more in the fish form and the lower lateral temporal cortex. We found OP4, and then we found some regions in the association or in the intermodal cortex. And of course, we found the interparietal sulcus, that is that region that is in between the motor cortex and the visual area. And this is kind of one way in which we were able to integrate the information coming from the task activation, the task connectivity, and the resting state connectivity. And we actually, to build this map, what we did is to normalize all the maps and then to do a sum of the maps, which is something that it is very intuitive, but in that way, we were able to integrate activation and connectivity to delineate where is the visual motor integration system. We can actually use different modalities to investigate what is happening in Alzheimer's disease. So as I was introducing, tau appears way before clinical symptoms and before the clinical diagnosis. We know that because of many investigations, both ex vivo and in vivo, we know, and also animal models, that in reality, tau first affects the locus coeruleus, and then after that, it will travel to the brain. We cannot measure with the current tau imaging, tau PET imaging, we cannot measure tau in the locus coeruleus. But we can measure tau in the brain, and we can use other imaging modalities as a proxy of what is happening in the locus coeruleus in the brainstem. In this research, we used anatomical MRI as a proxy to investigate how changes in the structure or LC were related to tau accumulation in the brain. We had around 75 individuals, and we have 2 images in both MRI and in tau PET. And what we did is to relate LC integrity, and LC integrity, so whenever inverted signal, so less integrity will mean more tau, like more tau is actually damaging LC. And we investigated how it related into the accumulation of tau across the entire cortex. We could have used many different graph theory metrics. For instance, again, if you have the different voxels that collected the information about the structural information of the LC, you can relate these different measures as a matrix to the matrix of each of the individuals that represents the tau PET signal. And what we observed when we did that is that the correlation or the connectivity between LC intensity and the mean of the tau signal was that whenever there was lower integrity of LC, we observed higher accumulation of tau years later. So we measure LC integrity at baseline, and we related LC integrity to tau years later. In this case, it was 3 years later, and we observed that tau was increasing in the areas of the brain that are normally related to the first appearance of tau in the brain at the cortical level. And to make sure that the pathway was in that direction, what we did is to actually extract the correlation values, and we did a computation in which we test the possibility of our hypothesized pathway that that tau damages first LC and then damages the medial temporal lobe. And we challenge our hypothesis against the reverse pattern in which tau will go from the medial temporal lobe into the LC. And we observe that the correlation values in our hypothesized pathway were higher than in the reverse pathway. And then what it was more interesting here is that our pathway was able to predict the cognitive decline of these individuals, meaning that individuals that had less or lower LC integrity, so they were accruing more tau in the LC, had more tau accumulation in the medial temporal lobe. So tau was traveling from the brainstem to the cortex, but they also had less or worse performance in the cognitive measurements. All in all, the use of different modalities in this case, and using the same principles that we learned from graph theory, help us identify the pathway in which tau is damaging the brain in individuals that are still in the preclinical stage of AD. This way, it's not only interesting using different modalities to resolve a question, but also using whatever we can take from graph theory and implementing it in other investigations. And with that, I will end that here. I would like to thank my mentor, Professor Jorge Sepulcre, and different members of the Sepulcre lab that have been in our lab. But also, we'd like to thank Dr. Heidi Jacobs and the Blue Spotters, which I did this last work that I was talking about with them. And I also would like to thank our funding bodies and the investigators and the patients and their family and their caregivers of the studies that I have using here, were the ADNI and the Harvard Aging Brain study. Thank you.